Good evening, everyone. My name is Jane Miller, and I am pleased to be the guest moderator tonight, standing in tonight for Kelly Gear-Ripken, National Chair of A Woman's Journey. On behalf of Johns Hopkins Medicine's A Woman's Journey, thank you for joining this evening for our monthly webcast series, Health Conversations That Matter. A Woman's Journey strives to improve your well-being through health education. The gut microbiome refers to the collection of more than 40 trillion bacteria or microbes that exist within the gastrointestinal tract. Each of the 1,000 species of bacteria in the human microbiome that plays a different role in your body. Most of them are extremely important for your health, while others may cause disease. Researchers are beginning to understand the impact of the composition of the microbiome and strive to identify the microbes or bacteria which are good and those that contribute to specific diseases. Tonight, we're pleased to be joined by infectious disease and cancer researcher, Dr. Cynthia Sears. In particular, Dr. Sears explores how to modify the microbiome to improve the detection of colon cancer and potentially a patient's responses to immunotherapy to treat cancer. Please use the Q&A on your screen to pose your questions to Dr. Sears, who will respond during the last 20 minutes of tonight's conversation. Our webcast tonight will conclude at 8 p.m. I want to take this opportunity to acknowledge and thank Johns Hopkins University's program, Hopkins at Home, for their production assistance. You can visit their website for additional lectures and courses throughout the year. And now I am pleased to welcome Dr. Sears.
Good evening, everybody. I'm delighted to be here. And as Jane just said, I'm Cynthia Sears. I'm an infectious diseases specialist. And so tonight I'm going to talk to you about the microbiome and your health. So I do have some disclosures. I have some research grants from industry, some of which are relevant to what I'm going to talk about tonight. I'm on an external advisory board, although it's not active yet, and I'm a writer and a reviewer for journals. So our menu tonight is I'm going to tell you what is the microbiome and why do we think the microbiome is critical to your health. I'm going to talk to you about major microbiome modifiers, and I'm going to talk to you about the microbiome in select diseases and how it may influence therapies, particularly immunotherapy, which is so important to cancer therapy at this time. I'm going to give you my take on yogurt and probiotics, and then I'm going to tell you why I think this is all exciting and what I think you should pay attention to. So our microbiome is integral to all of us. It's all around us. So every surface of our body is covered with huge numbers of microbes or bugs, as Jane just mentioned. And it's contiguous to any what we call mucosal surfaces inside our bodies. So the two words that are used most often are microbiome, which refers to all the genes in the organisms that we're colonized with, and then microbiota, which actually refers to the actual bugs. And my magic number is 100 trillion organisms that we share our bodies with. And as I said, they're on every external and then contiguous body surface into the mouth, into the esophagus, into the lungs, into the vagina, and so forth. So while the microbiome is made up of bacteria, viruses, fungi, and parasites, we know the most about bacteria because they're the easiest to work with. And we have three groups of bugs that we think about a lot. One are organisms that just stay with us. We call them commensals. They don't really interact with us and they don't really influence our health. The ones we care about the most, we call mutualists or symbiotes, and those are the ones that are communicating with our human cells. So we affect them and they affect us. And then there's a small percent that are those that can harm us and are called pathogens. So this slide looks complicated, but it really is a beautiful story about biology and is the beginning of our understanding how specific microbes can have such a profound impact within the microbiome. So the top left corner here I'm taking you back to 1970 when Dr. Hastings was in his laboratory and growing a bacterium in a tube. And when it got to a certain density, a density of which you could see all the bacteria in the tube, all of a sudden the bacteria glowed. And that simple observation actually led to an entire field in bacteriology called quorum sensing. And the importance of this is as bacteria get crowded together, they talk to each other. So we know that communities matter, and we know that bacteria in communities send out small molecules or chemicals that change the function of their neighbors and change the function of the community. Fast forward 20 years, and this paper published in Science was actually about this little animal called the Hawaiian bobtail squid. And we know that these little animals are in the seawater and they filter huge amounts of seawater every day. And the seawater is loaded with microbes. This organism has a light organ, and that's shown in the middle. These are scanning electron micrographs. And although all these bugs are flowing by the light organ every single day, only one bacterium actually sits down and stays put on the light organ. And you can see over about 3 weeks that there's a change in the morphology of the light organ. And at the end of that time, the entire organism glows at night. Now you might say, why? And it's a host defense mechanism. So the little animal floats in the water and its predators are below it, and it looks up, and the little animal looks like the night sky. So it protects the bobtail squid. And it turned out that the bacterium that sits down and creates the ability of this little animal to glow is exactly the same organism that Dr. Hastings was studying 20 years earlier. So here are some key concepts about the microbiome in health. So each of us has our own personalized microbiome. Each of us is quite different. However, all these bacteria we carry in our gut actually possess together similar functions in our health. And this is called the concept of functional redundancy. So the reason most of the time we're healthy is that we carry a group of bugs that actually are doing the same job for each of us. We know that different body sites have distinct microbiomes. So what's in the gut is very different than what's on the skin. And the skin is a particularly interesting example because the microbes that will sit on your arm are different than the microbes that are on your legs, or different than the microbes that are on your trunk. So they're specialized to work in the environments where they live. We know that the microbiome increases our ability to digest and absorb nutrients. So the microbiome is extremely important to our day-to-day health. And the microbiome is also essential to tissue development and normal immune function. So there's excellent data that suggests that the gut microbiome actually changes the function of the cells. It actually can induce the development of blood vessels in the gut. So it's extremely important to who we are. Now, because of this emerging data that how important the microbiome was to our basic function, the National Institutes of Health initiated a project called the Human Microbiome Project. And the key goal of this project was to understand what we call the ecology, just the baseline of how humans are colonized with all these microbes. And so they studied 250 individuals at 5 body sites over a period of time. And that is really the baseline data from which we continue to work today. And in the process of doing that work, we learned that the greatest concentration of microbes in the human body is in the gut. Second is the skin. So how can we modify our microbiome? Well, there are 3 major factors. One is diet, the second is medication, and the third is our genes. I'm not really going to talk about genes today, although I'd be happy to entertain questions, but the genetics that the host DNA that we each carry with us has a continuous pervasive effect on our microbiomes, but it's a little hard to detect, so it's not as well studied. Now, the number one modifier of the microbiome is likely diet. And we began to understand that in the 1930s when germ-free mice were developed. And what was observed was that germ-free mice eat continuously, but they don't gain as much weight. If they're given a gut microbiome, then they slow down their eating patterns, but they gain weight, which is just proof of the importance of these microbes for our guts to be efficient for our health. Now, on this slide, I have 3 studies from the high-level journals, but each had a particular message that I found particularly engaging and important. The first study in 2011 studied what happens when you change a diet. So if today you're a vegan and tomorrow you become a meat eater, what they were able to show was that in 24 to 48 hours, your gut microbiome switches around. The secondary message from that is we each in our gut microbiome probably have the power by what we eat to switch the function and make it more healthy for us as versus less healthy. In the second paper down here in 2015, this was a fascinating study done by the University of Pittsburgh. And what they did was they studied the diet and metabolic effects of the diet in African Americans who were on a low fiber, high fat diet, and Africans in Africa that were on a high fiber, low fat diet. After they established that baseline, they switched the diets. And they switched the diets for only 2 weeks. And in that, just after 2 weeks, the metabolism of the African Americans improved on a high fiber, low fat diet, and the metabolism of the Africans deteriorated on a high fat, low fiber diet. So again, it just illustrates how quickly food can affect how healthy we are. In the third study, which was sponsored in part by the Israeli government, a very, very extensive study. But the message I took away that I found really informative was the fact that one person who eats pizza can have absolutely no impact on their metabolism or their blood glucose. Yet another person eats the same pizza and their blood glucose may go up. So we each have personal features that influence our response to diet, and that's likely embedded in our genes. The second most important microbiome modifier are medications. And among medications, although there are now many that have been shown to modify the microbiome, the most important is antibiotics. And virtually everyone on this call, I think, has probably taken at least one course of antibiotics in their life. There are many antibiotics prescribed. In the infectious diseases world, we worry about the inappropriate use of antibiotics because of the consequences of that. We know abroad there are what we call over-the-counter antibiotics. You don't need a doctor's prescription to walk into a pharmacy and get whatever antibiotic you would like. And we know that antibiotics have been used in development of our food or growing our food. So there is no question when you need antibiotics, you should take antibiotics. Antibiotics save lives, but they can have adverse outcomes. And the one we're worrying about the most is antimicrobial resistance, which is happening across the globe. And there is a paucity of development of new antibiotics. So this is a grave concern. to the infectious diseases field. A second is an infection you've probably heard of called C. difficile. So when you take antibiotics, you deplete your gut microbiome, and this particular organism can bloom in the gut and cause diarrheal disease and a lot of abdominal pain. There are allergic reactions. Nausea, vomiting, and diarrhea are very common with taking antibiotics, as is Candida vaginitis. The one effect that's less clear, but which I worry about, is that we used to say that if you took a course of antibiotics, your gut microbiome would be back in a couple months. Everything would be back to baseline. It's now not so clear that that's correct. So there are now studies that show that there's this subtle, persistent change in the microbiome after taking antibiotics. And for example, distant antibiotic use has been identified as a potential risk factor for increasing the likelihood of getting colon cancer. In a study we did, antibiotic exposure 10 years before the appearance of colon cancer appeared to influence the frequency with which colon cancer occurred. It's not proven, it's an association, but it is a worrisome association. So at a deeper level, we worry that this exposure and sort of this subtle remodeling of our microbiome that goes on through our life may be contributing, for example, to other what we call non-communicable diseases, things like autoimmune disease and others. So microbiome diseases, I would call the frontier, and this is an exciting field. So every body site is under study. Some of the most interesting data relate to systemic and therapeutic induced diseases such as obesity and malnutrition, diabetes, hepatitis, atherosclerosis or heart disease, and so on and so forth. And really every cancer, or many of the major cancers, are also being studied. So we know that single microbes are responsible for many cancers. In fact, about 25 to 30% of cancers across the globe are associated with a chronic infection by a single microbe. In the case of stomach cancer, it's Helicobacter pylori, that accounts for about 90% of gastric cancer, and about 10% is thought to relate to chronic infection with Epstein-Barr virus. Liver cancer is linked to hepatitis B and C. Cervical cancer to human papillomavirus, as is anal or penile cancer. Bladder cancer in Southeast Asia is linked to schistosoma hematobium, that's a parasite, as is biliary cancer, but different parasites as shown here. So we know that single microbes can be strongly linked and causal in various cancers. Now, as was already said, I've worked for many years now trying to look at the relationship between particular bacteria and colon cancer. Obviously, the gut has this massive community of bacteria, and it's an obvious environmental risk factor in the gut for colon cancer. So we've been interested in trying to unravel what is important. And I use this slide to illustrate the type of thinking that is happening across any cancer. So on one hand, we're concerned about specific organisms, as listed here: Fusobacterium nucleatum, PKS positive E. coli, enterotoxigenic Bacteroides fragilis. These are particular organisms that contain virulence factors, what we call virulence factors, that can be can promote cancer in the gut. And this picture illustrates that. So in the control animal, these are mice guts. You don't see any of these big, dark, black tumors. However, if we add this single bug, the enterotoxigenic Bacteroides fragilis, to the complex microbiome of the mouse, what we see about 12 weeks later are tumors in the gut. Importantly, each of these bacteria have now had a substantive association with colon cancer in humans. Now, at the other end of the spectrum is what I talked to you earlier about, Dr. Hastings and the community function. And that's the other piece of this puzzle, is the community. How can it contribute to disease? And one thing we've been investigating is what are called biofilms. And this cartoon illustrates what a biofilm is. So the entire gut that we each have, our colon has this layer of epithelial cells. And above that is this light gray, is called the inner mucus layer. And that mucus layer is very dense. It's a host defense mechanism. It's sterile in the gut. It's preventing all the bugs from getting at our epithelial cells. However, this can break down. And when it does, you can see this dense invasion of that supposedly sterile inner mucus layer. And we've been able to show that these bugs then change the function of the epithelial cells in the direction of cancer. Studying these biofilms has also enabled us to identify new contributors to colon cancer. It's bugs that are associated with colon cancer. Now, this is a list of all the cancers that are being pretty intensely studied. And all, as you can see, are in places where the microbes exist and where there's exposure to the environment. The one exception is the liver. Here, it has a somewhat distant association to the outside world, but there's a circulation between the gut and the liver that happens continuously, which is why the liver is exposed. Now, on this list, as I wrote here, the list is longer than the evidence. So the studies in these areas are at an earlier stage than what I just described with colon cancer. I did want to point out the important work of Dr. Dipali Sharma's lab here at Hopkins, looking at the linkage between breast cancer and particular microbes. Now, I mentioned that we're concerned that the microbiome is one feature that holds back the success of immunotherapy. As you all know, cancer immunotherapy has been the greatest breakthrough in cancer therapy in probably 30 or 40 years. But only a minority of patients, for the most part, there are some exceptions, respond to cancer immunotherapy. On the overall, it's only up to about 20 to 30% of individuals who will respond to immunotherapy when used as part of their treatment for cancer. This slide just shows some of the very early studies. You see, they just happened in 2018, indicating that there could be a linkage between the microbiome and success of immunotherapy. Now, that has led to a lot of work trying to understand which exact microbes in different cancers are most important and what we can do to probably change the microbiome and perhaps enhance the responses to immunotherapy. So when we talk about changing the microbiome, I've already introduced diet as a very important feature. And I've mentioned medications, and then I'm adding to that supplements. Supplements would be something like what we call a prebiotic or a probiotic, and I'll come back to that. There are fecal microbiota transplant. And what that means is the stool of a person who has, for example, responded to immunotherapy has now in early trials been put into someone who's failing immunotherapy. It's introduced by colonoscopy. And there has been some individuals who appear to respond to this, but it's still trying to be unraveled exactly which communities and which organisms are most important. A really exciting feature is many biotechs are now trying to create engineered microbes that can be taken and which would change the function of the microbiome. And this is an exciting area of research. So I wanted to mention probiotics, which we also call dietary supplements and medical foods. And the definition of a probiotic is the ingestion of live organisms, which when given in adequate amounts confer a health benefit on the host. A prebiotic is a supplement that enhances the growth of beneficial gut microbes. So probiotics come in many shapes or forms. So all you have to go is to some of the GNC stores and the like. You'll see probiotics made up of single bacteria, multiple bacteria, different types of bacteria. It is really important for you to understand that there is no quality control in this industry, and there is indeed no regulatory oversight. Years ago, these were classified as supplements, and so they are not FDA regulated or evaluated. They fall under a category called GRAS, which stands for generally regarded as safe. Because of this lack of rigor in this field, we actually have few very well done studies. And in fact, when this was recently looked at, only 2% of the studies actually reported safety measures in the development of probiotics. At this point in time, there is no FDA approved product for any condition that no probiotic is FDA approved. So it is not proven that these help in any condition. And there's emerging data that suggests they may do harm in certain conditions. So the evidence is limited, but there is at this point no support for probiotic supplementation that is able to improve outcomes with cancer immunotherapy. And this paper shown here, published in Science just 2 years ago, is a major evidence that gets cited. And I just highlighted the most important sentence, and that was that amongst patients treated with immune checkpoint blockers or ICBs or immunotherapy, the most pronounced benefit was observed in patients who took sufficient dietary fiber intake and no probiotic use. I also want to mention the potential health benefits of yogurt and kefir, which are really ancient foods. So as you know, all yogurt has 2 bacteria in it that actually is what creates the yogurt, and those bugs are listed here. When manufacturers add additional live bacteria, then these are called probiotic yogurts. Kefir, in contrast, is actually a fermented milk drink that's actually quite complex and generally contains both live bacteria and yeast, although the exact formulation is not clear. At least it's never been clear to me from anything I've read or what I've looked at on labels in the grocery store. Yogurt is a nutrient-dense food that is thought to perhaps facilitate calcium absorption. So in contrast to probiotics, yogurt and kefir are considered safe foods. And kefir has been studied, for example, in a limited way, very limited, in individuals with C. difficile disease, and has appeared to be helpful in that condition. Certainly, these are not harmful. So here are a few take-home messages for you. This is, the microbiome field is really quite new. It's 20 years old, but in science, that is a short period of time. And as you can see, there's so much complexity to this field, both in trying to understand how the microbiome contributes to health, and then how it's contributing to disease, that there is much work to do, but it is definitely a field of promise. So I would suggest to you that it's important to try to protect your microbiome. eat a healthy diet. And by that, I mean the US recommended diets, Mediterranean diet, DASH diet, are all very reasonable diets for each of us to adhere to. Exercise appears to be helpful as well. So there's now data showing that exercise modifies our microbiome, and in both hereditary colon cancer and breast cancer, there are studies that are being conducted in the United States, Europe, and Australia that actually show that there's improvement in how individuals with cancer feel and how they do clinically. They do better when they exercise. I suggest that if you're prescribed antibiotics, you make sure you understand why you're taking antibiotics. And remember that antibiotics don't treat viral infections. Probiotics are not yet of proven benefit, but yogurt and kefir offer low-cost foods that may benefit health. And I would say times are pretty exciting in this field. In 2023, we now have 2 FDA-approved microbiota modulating therapies, one called Rebiota and the other called Voust. Rebiota is a quality-controlled fecal microbiota therapy, and Voust is an oral microbiota therapy that is designed to try to replace bad microbes with good microbes in your gut. Both were developed for treatment of what we call recurrent C. difficile disease. So when C. difficile occurs, it can come back. And so after it has come back a few times, these are potential therapies that can be used. And with that, I will conclude and be happy to answer any questions.
Thank you, Dr. Sears. Let's start with just one question. Has there been any research that's identified specific microbes that have been identified as a cause of other cancers or diseases?
Such as other than the ones I mentioned?
Yes, for example, the Dr. Sukumar's research identifying microbe as a cause of breast cancer.
So it's Dr. Sharma that has done that work. And I wouldn't say that we can say there's a microbe that causes breast cancer. What has been found is when people have been studying the microbiome of the normal breast, they identified different organisms that were present. Obviously, there's a connection between the skin and all the ductals that are in the breast. And what Dr. Sharma's lab did was look at the available data and identified that an organism called Bacteroides fragilis was there pretty commonly. And so we worked with them. They took our strains of enterotoxigenic Bacteroides fragilis and tested them in animal models to see what their impact was on the biology of breast cancer cells, and identified that when the mouse was colonized with these enterotoxigenic Bacteroides fragilis, but not by strains of Bacteroides fragilis that didn't make the toxin that makes these organisms more virulent, that with this particular protein called the Bacteroides fragilis toxin, when that was made, it led to proliferation of the cells. It increased the rate of metastasis. So human studies are now starting to take a deeper look at this, but we don't have all the data as yet.
We have a viewer question from Sheila. Can the microbiome be a contributing cause of depression and IBS and other gut-related issues? And what type of physician should patients seek in trying to modify their microbiome or mitigate medical problems?
So those are great questions. I think we think that there are probably contributions of the gut microbiome to things like, certainly to inflammatory bowel disease, but also irritable bowel disease, which is what IBS is, that the microbes may be influencing the nerves in the gut and impacting how the gut functions. You asked about depression. This is also a very exciting area, and it's not just depression. It's diseases like multiple sclerosis, Parkinson's disease, even Alzheimer's are now being studied for linkage to the gut microbiome. There's an axis called the gut-brain axis, because there's nerves that come from the brain that come all the way down to the gut. And it's thought that there's communication going on and potential molecules being made that are influencing these disease processes. Now, there isn't a microbiome doctor yet. And in fact, for the general health, there's no microbiome test that is going to be helpful at this point in time to a physician. We don't know enough to be able to say, okay, we're going to do that test and we know what should happen. However, that's why I think my general advice is to eat a healthy diet, exercise. They sound like really simple things, but they really do make a difference when we do them consistently. Don't smoke. Smoking affects the microbiome. Smoking is a risk factor for colon cancer, and the data suggests that it increases the impact on cancers in the right side of the colon.
Yeah, this is kind of a really simple question, but food is a big part of this. Am I correct? It's like you are what you eat,
Yeah,
and what you ingest.
That's right. That's why I emphasize that, because we're each in control of what we eat, or we try to be in control of what we eat. And, it is a very important thing that you can do to be healthy.
We have a question from Kathy. What are your thoughts on berberine? Am I pronouncing that correctly? Berberine? To improve gut health.
So, it's been years since I actually read about berberine. I don't think it's likely to be helpful to most people. I haven't seen anything recently, which is why I don't know any recent data, but just like probiotics, most of these things, there's been interest in curcumin related to turmeric, and that failed in clinical trials.
A question from Rachel, which is a food question. What if you eat certain foods and get bloated as a result? What causes that? Is that your microbiome rejecting what you're eating? And what can you do to reduce the bloating?
Yeah, that's a great question as well. Each of us has a unique microbiome, and some people are, shall we say, gassier than others. So I do think we have to pay attention to how particular foods affect us. I have a set of foods I sort of steer away from because they cause gut distress. I assume that your particular microbiome is digesting in a way that's making you gassier, and that's the reason. When that gets severe, I think really laying down and letting time pass is about the most we can do.
Question from Nicole. What types of laboratory tests do you recommend to check the health of one's gut microbiome?
So I don't recommend any.
Is it possible?
It's not really possible. There was a big study called the American Gut Project, in which I think for $100 you could send in your stool, and you can find companies that will do your microbiome analysis. But we don't know what to do with that information at this point in time. So I don't encourage people to do that. I have gotten calls, people have done it, and they want a pronouncement, right, of what those results mean. And we in the field are still debating what is a healthy microbiome, or what is a normal microbiome? And I think the answer goes back to what I mentioned, that each of us have a unique microbiome for the most part. If you get 100 people in a room, there's going to be a few organisms that are going to be found in more than one person. So I think the answer is that functional redundancies. There's so many possibilities that it's difficult to pin it down to just a few.
Well, we have a couple of questions that kind of related to that. Is anyone studying the plasma role of the gut biome in food intolerance?
So the answer is, I would say yes. I mean, there are certain food intolerant conditions, such as celiac disease, that we've gained increased appreciation for, and that different people have different sensitivities to gluten. Some people it's fine, but a number of people without true celiac disease do find that if they go off gluten, that they feel better, and that they are having a reaction to the gluten.
Is this related to the gut microbiome, the food intolerance? I mean, a lot of people really suffer from different foods that really bother them.
Right, right. And I don't know the answer to that specifically. As I said, we each need to pay attention to our bodies. And I'm gonna guess that some mix of your genes and your microbiome and specific foods may yield these outcomes, but we don't know how to pin it down at this point in time.
A question from Debbie. How do you how does the biologic affect the microbiome? How does the biologic help or not help in Crohn's disease?
Biologics being used to treat inflammatory bowel disease, is that?
Presumably, yes.
Yeah, so inflammatory bowel disease is probably our best example of a disease that is driven by your host genes, the microbiome, and your immune system. And I say that because now there are over 200 what we call polymorphisms in genes. So just subtle changes in the DNA structure that leads to a slightly modified protein. And those polymorphisms are important to inflammatory bowel disease. As a result of those gene changes, the makeup of the microbiome can shift, and then there's a feedback loop to the immune system in the what we call the submucosa, or under those epithelial cells that I was showing you. So there is a very complex disease, and each person can have different gene mutations. And I have to say, it's one of the difficult parts of IBD. There are many, many therapies, biologics that are used. People will respond to one, but they won't respond to another, or they fail one and they move on to another. And usually we don't know the exact reasons why that happens, but that is a very active area of research, both here at Hopkins and many other places. I think it is one of our best examples and common examples. IBD is not an infrequent disease, and it's a disease that we know is increasing across the globe. So it is thought to be this mix of your host genes, the microbiome, and your immune system response.
A question from Michelle. Are there any studies going on to study the influence of our microbiota on vagus? Is it vagus or vagus? Vagus nerve.
Yeah, the vagus nerve.
Vagus nerve and gut-brain communications.
There is a lot of work going on in that area that I think is really fascinating. So I don't know that area as well as the colon cancer field, but I do pay attention, have been very impressed how the data are growing in strength. about this connection between the gut and the brain. And the vagus nerve is the nerve I was referring to that comes all the way down from the brain and goes all the way down to the gut, and is a very important nerve that controls how the motility in our gut, in part.
Question from Preeti. Is it true that bacteria, fit a mucus?
Mucus?
Yeah, FITMICUTUS.
Fit a mucus.
It's associated with ovarian cancer.
You got me. I do not know that particular compound. I'm sorry.
Okay, and it goes on, if someone has platinum resistant ovarian cancer, is there anything in altering the gut microbiome that could help?
Not to my knowledge. I think those types of cancers are among those that are not studied very well as yet.
This is another food question. Kombucha?
Oh, kombucha.
Drink?
Yes.
Helpful to the microbiome?
So it is said. I don't know chapter and verse, but it is thought to be, by its nature, a positive food for us. And actually, they now sell it in the grocery stores. It's so funny.
A lot.
A lot. Yeah, I personally just recently said, I'm just going to buy some and try it. And actually, it's a pretty low calorie substitute for soda. It's very interesting. It has a very similar taste, but it's low calorie, actually.
Another question. What happens if a baby is born through C-section and doesn't go through the vaginal canal?
Yeah, this is a great question, and also an area of intense investigation. So the microbiome of a newborn is seeded by vaginal delivery, and that creates what we think of as a normal microbiome in a baby. When a baby is born by C-section, the microbiome comes out different because of exposure to the mother's skin, not going through the birth canal, and a greater exposure to hospital organisms. And there has been linkage now between C-section deliveries and certain early childhood diseases over the next few years. So trials are going on where the mother, if a C-section has to happen, that's certainly happen medically, that swabs are put into the vagina of the mother, and the baby is covered with the vaginal flora from these swabs as soon as the baby comes out, put in their mouth, their nose, their face, rubbed all over them to try to force, if you will, colonization that's considered more natural. And we don't know yet whether that approach is going to mitigate some of the subsequent illnesses like increased allergy, asthma, or some of the linkages that have been reported. I wouldn't say they're causal, right? But these are associations that have been seen.
A question from Rachel. What is the timetable for microbiome research to be put into practice to help with disease?
Another great question. So we have 2 therapies that were approved this year that we're excited about. We're a little unexcited by the cost and our concerns that insurance may not be fully supportive. But we're very excited that they're on the market because they look like better alternatives than some of the things like garden variety, if you will, fecal microbiota therapy, where you're taking a stool from an average person and putting it in another person. and that seems less safe than taking a defined product, which is what Rebiota is, or taking the oral product, which is what Voust is. So we are excited about that. There are trials now being done with the engineered microbes that I mentioned, or probiotics, to see if we can change the course of disease. For example, we're hoping to participate in some of these here at Hopkins, in which they'll be done under controlled conditions, and patients will be followed very carefully for safety and for changes in their microbiome, changes in their metabolism, that to see if it's helpful to their therapy.
Question from Erica. What can someone do daily about constant and consistent constipation?
That is, again, a difficult problem. It depends on the individual. I mean, one simple thing that can be done is something like a Metamucil or a Metamucil-like product. There are generic versions of that, where you're dialing up the fiber, which can sometimes be helpful. The other general recommendation is often drinking more water, so the stool is better hydrated and therefore easier to pass. There are other possibilities, and for some people this is a very serious problem, but those are the two simplest courses to take. Now, a lot of people don't like taking Metamucil. It's hard for them to swallow, but it is one approach that might be helpful.
Is there, I think you probably have answered this in a couple of different, questions that have been asked. Is there anything that can be done to alter the composition of our microbiome?
Yeah, if I was at a meeting once, and a scientist had measured his microbiome every day for 3 weeks. So he analyzed it with fancy techniques every day for 3 weeks, and showed all the variability that he saw, which is undoubtedly linked to diet. So our diets are very powerful. So what can happen with a diet is an organism that's there in such low frequency, we can't detect it. After you change your diet, can bloom, and now we can detect it. So I always think of that as empowering for us, that if we choose to eat a healthy diet, in us are the right microbes to probably help us in terms of our general health.
A question which you just discussed about the C-section delivery. Can you elaborate on the childhood diseases related to C-section delivery?
Yeah, the ones that I'm most aware of are the allergic diseases, things like eczema that may occur in young children, and a greater frequency of asthma and other allergic type diseases.
I'd like to ask you a question we were talking about before we started tonight, which has to do with colon cancer and the increase in younger people that are being diagnosed with colon cancer.
Yeah, this is a very serious problem, so that we are now seeing for several decades an increase that was initially not so clear, but now it is clear that there's increased colon cancer and rectal cancer occurring in individuals under the age of 50. As many of you may know, the guidelines for getting a colonoscopy to prevent colon cancer shifted in the last 2 years from age 50 to age 45. Yet we are seeing people in their 20s and 30s with these illnesses that we have previously thought would happen predominantly in older individuals. And overall, they are still predominantly in older individuals, but about 50 new cases of colon or rectal cancer are being detected per day. in individuals under the age of 50, which is outside preventive guidelines. So there's a lot of work going on both at the National Cancer Institute and across the country by foundations such as Fight CRC or the Alliance for Colon Cancer that are trying to understand what has happened. And it's thought probably to be environmental influences that occurred with birth cohorts. So things like, we know that in the late 1990s to the early 2000s, we had an increased use of antibiotics. We know that we had the onset of an increase in obesity in this country. And both of those are factors that have been linked to colon cancer. So something has changed to change the fundamental epidemiology of colon cancer. And it's not unique to the United States or Europe. It's actually across the globe. So in Tanzania, I have colleagues who have begun to recognize that about 40% of their colon cancer is now occurring in young people. And it was a disease that was previously rare in the countries with lower socioeconomic conditions.
Are there, when we talk about prevention of colon cancer, obviously diet, et cetera, but at that younger age too, am I correct?
Correct. There's now data suggesting that, for example, I saw this just the other week, lipid abnormalities in adolescents are going to impact their likelihood of heart disease, for example, later. So the principles of a good diet and exercise actually stretch back to good habits starting in childhood.
This might be our last question. We're getting close to 8 o'clock. From Carol, could an increased fiber intake and subsequent increased variety of the microbiome mitigate the disease-producing microbes in the small intestine in conditions such as SIBO?
Small intestinal bowel overgrowth is what SIBO stands for. So it turns out that the greatest concentration of organisms in our gut is in the colon. There is much smaller populations in the small intestine. And SIBO tends to happen when there's a blind loop or there's some anatomical abnormality in the upper gut, or when there's the motility of the gut is abnormal. That said, that condition is linked to microbes in some cases. So the answer is, I guess, yes, but I don't know that diet will do it there as much as the diet can help your colon.
Okay, one more question. Is there anything accepted as a normal bowel movement rate? What rate is typical?
That is extremely variable person to person. The magic number in the GI field is 200 grams or 200 milliliters of stool per day. Now, whether you excrete that in one sitting or multiple sittings is really individual dependent. What you should not be doing, though, is getting up in the middle of the night. If you're getting up in the middle of the night because of bowel urgency, that's not abnormal. That's not normal.
All right. I'd like to thank Dr. Sears tonight for speaking with us. She's graciously agreed to respond to any unanswered questions that were asked this evening. In a couple of weeks, you'll receive an email outlining the outstanding questions and answers. In the coming weeks, a video of tonight's live stream will be available on the Woman's Journey website, hopkinsmedicine.org/A Woman's Journey under videos on demand. If you have enjoyed tonight's discussion, please check out our website, hopkinsmedicine.org/A Woman's Journey, for more information about upcoming webinars, our podcasts, and our monthly email. Good night and stay well.
Thank you.